Hi everybody and welcome to our module on virulence. First let's talk about virulence factors. So these are features of bacteria that allow evasion of host defenses. And there are lots of virulence factors that have been identified for various bacteria, but three that you definitely should know, especially for USMLE Step 1, are protein A, IgA protease, and M protein. Protein A is a key virulence factor for Staph aureus, and you can just think of this A in protein A as standing for aureus, for Staph aureus, and it's part of the peptidoglycan cell wall of Staph aureus. And what protein A does to help the bacteria invade the body is inhibit phagocytosis. The way it does this is by binding the Fc portion of IgG antibodies. So IgG antibodies have a straight portion, which is called the Fc portion. That stands for fragment crystallizable. And then they also have a branched portion that looks something like this. That's called the Fab portion for fragment antigen binding. And normally when you have a bacterial cell like this, what happens is the antibody comes in and binds the surface like this with the Fab portion. And then this draws in complement and other immune system cells to kill the bacteria. What protein A does is it turns the IgG molecule upside down. So if this is a Staph aureus cell, it's going to have this protein A sitting on its surface here, and it's going to bind the Fc portion right here, and this turns the antibody upside down, and therefore it's not able to attach to the cell and activate the immune system. And this is part of the way by which Staph aureus bacteria can avoid immune destruction. So through binding of the IgG antibodies upside down, protein A will prevent opsonization and phagocytosis by macrophages, and they're also going to prevent complement activation. And the net result is that this helps the bacteria avoid the immune system altogether. IgA protease is an enzyme that cleaves the IgA antibodies, and recall that the IgA antibodies are very important for mucosal immunity. So this protease cleaves those antibodies and allows the bacteria to persist on mucosal surfaces so that they can colonize the mucosal surfaces. And this is a very important virulence factor for strep pneumonia. It's part of how strep pneumonia is able to get into the respiratory tract and into the lungs. It's also important for H. influenza, and it's also important for Neisseria. Both gonorrhea and meningitis have IgA protease. The M protein is an important virulence factor for group A strep, which is also called strep pyogenes. It's a surface molecule found on the bacteria, and you'll recall that group A strep is responsible for strep throat and the sequelae of strep throat like rheumatic fever. The M protein works by preventing phagocytosis, and it does this largely by interfering with the complement system. First of all, the M protein can bind factor H. I described factor H in more detail in the immunology modules, but it's a factor found in our own serum that protects our own cells from complement activation, and the M protein can hijack this factor and use it to protect the bacteria. In addition, the M protein has been shown to break down C3 convertase, and this prevents the formation of C3b on the surface of the bacteria, and C3b is a very important opsonin for phagocytosis. The M protein shares many properties with myosin, and it's believed this may be the basis of rheumatic heart disease, where there's autoimmune damage to the heart in patients who have had a bacterial infection with strep pyogenes. In addition, the M protein is important when it comes to the post-strep complications. So patients who have had a strep throat can develop rheumatic heart disease or glomerular nephritis following the infection, and it turns out that different M protein subtypes are associated with each complication. It's very interesting. Patients almost never get both rheumatic heart disease and glomerular nephritis. They get one or the other, and that's because different M protein subtypes are associated with each complication. Now let's talk about bacterial toxins. So these are substances that come from bacteria that are toxic to the human body, and they're generally classified in two groups. The first group is called endotoxin, and the second group is called exotoxin. And even though the names are similar and sound alike, they're very, very different. Endotoxin is a substance that's only found in gram-negative bacteria. It's a component of the outer cell membrane of gram-negative bugs, and recall that only gram-negative bugs have an outer cell membrane. Gram-positive bacteria do not. And it's made up of a substance called lipopolysaccharide that we'll talk about in a minute. Exotoxins are proteins. They're synthesized by some bacteria and secreted, and when they get into the body or into the bloodstream, they can cause illness. Let's talk about endotoxin first. So as I said before, it's a component of the gram-negative bacterial cell wall. It's released into the tissues and the bloodstream when bacteria die. It's not secreted. This is an important distinction between endotoxin and exotoxins. Exotoxins are secreted by bacteria that make them. Endotoxins are actually part of the cell wall, and the way they get into tissues in the bloodstream is when the bacteria die. They're made up of lipopolysaccharide complexes called LPS, and this is also an important distinction between endotoxin and exotoxins. Exotoxins are proteins. Endotoxins are made up of fat, that's why they have the lipo in the name, and they're made up of sugar, that's why they have the polysaccharide in the name. There are many different variants of endotoxin found in different gram-negative bacteria, but they all share some common features. They're all made up of a core that contains a substance called lipid A, and lipid A is what's responsible for most of the toxicity. It triggers the immune system, and the secretion of cytokines and the activation of cells leads to hypotension and edema and lots of the illness features of patients who are infected with bugs that carry endotoxin. Endotoxins also have an O antigen. This is on the end of the sugars, and it's different based on the bacterial strain you're talking about. The O antigens do not cause disease by themselves, so we know that they are not part of what makes people sick, but they are a target for the immune system, and they are part of the way that antibodies and other immune system cells target the endotoxin and are able to clear it from the body. Patients who are infected with gram-negative bacteria can get very sick, and that is because of the endotoxin. Endotoxin can lead to fever and shock. It triggers the release of TNF and IL-1, and these are very important immune components in the syndromes of sepsis and septic shock. So part of the reason patients who have sepsis or septic shock get hypotensive and tachycardic is because of the release of these mediators, and their release is triggered by endotoxin. Unfortunately, endotoxin generates a weak antibody response, so you can't vaccinate against it. If you could, it would be wonderful. You could essentially prevent many gram-negative infections, or at least prevent them from getting so serious, but this isn't the case because it doesn't generate a strong antibody response. And then another feature of endotoxin you should know about is that they are called heat stable, and this means they can tolerate relatively high temperatures. So for example, when a patient gets febrile, it doesn't kill off the endotoxin because it's able to tolerate those higher body temperatures. There's a very high yield tie-in between microbiology and immunology when it comes to endotoxin. There are a number of ways that endotoxin and lipopolysaccharides can activate the immune system, and the board examiners like to go after this to see if you can understand how bacterial infections trigger the immune response. So make sure you understand all the things on this slide about how the immune system is activated by lipopolysaccharides. So first of all, lipopolysaccharides can activate macrophages, and they do this by binding to the receptor called CD14. And it's high yield to remember CD14, that's called the endotoxin receptor. Once these macrophages are stimulated through CD14, they can release IL-1 and TNF, which you should know are cytokines that are released by macrophages. IL-1 can cause fever, TNF can cause fever and hypotension. These macrophages have also been shown to release nitric oxide, which as you know is a vasodilator and can lower the blood pressure. Endotoxin and LPS can also activate the complement system, and when they do, complement C3A and C5A get released. C3A, you may know, is called an anaphylotoxin because it's responsible for anaphylaxis. It can cause hypotension, and it can cause increases in vascular permeability and edema. C5A is also an anaphylotoxin, and it can draw in neutrophils by chemotaxis, and it can also cause edema. And then finally, endotoxin has been shown to directly activate tissue factor, and this can lead to coagulation problems and disseminated intravascular coagulation. So make sure you understand this very important tie-in between bacterial infections and endotoxin and immunology. There are some gram-negative bacteria that do not have endotoxin made up of lipopolysaccharides, but instead their endotoxins are made up of a compound called lipo-oligosaccharides, or LOS. These are very similar to LPS. They're still endotoxins. They still trigger an immune response. They just have some structural differences. For example, they generally lack the O antigen. And these LOS are found on non-enteric gram-negatives. And the most important example is Neisseria meningitidis. So Neisseria meningitidis can get into the bloodstream and cause shock and make people sick. And it does this through a typical endotoxin reaction, but the endotoxin is not LPS. Instead, it is lipo-oligosaccharide for its endotoxin. So anyone who's infected with a gram-negative bacteria can potentially develop a systemic reaction to endotoxin. But two classic examples that you should know about are meningococcemia and gram-negative sepsis. So when people have meningitis from Neisseria meningitidis and then the bacteria gets into the bloodstream, they can get very, very sick. They can get hypotensive and go into shock, and that's related at least in part to the endotoxin. Also, patients who have sepsis from a gram-negative bacteria can get very sick. So for example, if someone has a urinary tract infection and the E. coli, which is a gram-negative bacteria, gets into the bloodstream, they can develop gram-negative sepsis, which is also a very serious condition. There's often shock and hypotension, and this is also related to the endotoxin. Now let's talk about exotoxins, and these are very different from endotoxins, even though the names sound similar. Exotoxins are proteins, and they are secreted by bacteria, and the exotoxins then enter the body and they cause disease symptoms. The classic exotoxin has a two-component structure called an AB polypeptide. So one protein is called the A component, and A stands for active. This is the component that causes illness and disease. The other component is called the B component, and B stands for binding, and this is the part of the exotoxin that binds to cells. And most of these bind to cell surfaces, sort of like what I've drawn down here, and then they're internalized inside the cell, and once they enter cells in the human body, they cause disease. There are lots of different mechanisms for how the B portion of the exotoxin binds a cell and gets inside. So there are many known exotoxins, and they all have lots of different toxic effects on the body. But in general, you can categorize the toxic effects of endotoxins into a couple of general categories. There are those that inhibit protein synthesis in cells. Some of them increase fluid secretion, and most of the exotoxins that do this cause diarrhea. They cause fluid to be secreted into the GI tract. Some exotoxins inhibit phagocytosis. Some block neurotransmitter release, and these are toxins like botulinum toxin that cause muscle weakness. Others lyse cell membranes, and then finally there are superantigens, which are the last exotoxins that we'll talk about. So we're going to go through all these toxins by the mechanisms through which they cause their toxic effects. And the first mechanism we're going to discuss are toxins that are protein synthesis inhibitors. And toxins that do this include diphtheria, Pseudomonas, Shigella, and enterohemorrhagic E. coli or EHEC. So the first two protein synthesis inhibitor toxins are the toxins that go with diphtheria and Pseudomonas. And both of these work by a similar biochemical mechanism. They both work by adding a molecule called ADP ribose to proteins. And once this ADP ribose has been added, the protein becomes dysfunctional. And this is how these block protein synthesis. So let's talk about diphtheria toxin in a little more detail. So recall that Corynebacterium diphtheria causes a sore throat with a membrane in the back of the pharynx and swollen lymph nodes. And you almost never see diphtheria infection anymore because it's been largely eradicated by vaccination. There's something called the DPT vaccine that children receive, which protects them from diphtheria infection. Diphtheria toxin works by ADP ribosylation, and an ADP ribosylates something called elongation factor 2, and EF2 is necessary for protein synthesis inside cells. So I've drawn the reaction that occurs down here at the bottom of the screen. So the toxin stimulates this reaction so that NAD will be converted to nicotinamide, and as part of that, an ADP ribosol group will be transferred to elongation factor 2, and then no further protein synthesis can occur. And this is a very lethal toxin. It only takes a tiny amount of toxin inside the cell to completely shut down protein synthesis. Pseudomonas makes an exotoxin that works in a very similar manner. Recall that Pseudomonas aeruginosa causes many types of infections, including skin infections, sepsis, and pneumonia, and I talk about this in detail in the module on gram-negative rods. But Pseudomonas makes an exotoxin called exotoxin A, and this has the same mechanism as diphtheria toxin. Exotoxin A, ADP ribosylates elongation factor 2, and this blocks protein synthesis. Next, let's talk about Shigella. So Shigella secretes a toxin that interferes with protein synthesis, but it does so through a different mechanism than ADP ribosylation. So recall that Shigella causes infectious diarrhea, and it makes a toxin called shigatoxin. And what shigatoxin does is it binds to the 60S ribosomal unit in cells to interfere with protein synthesis. When shigatoxin binds this unit, it removes a very specific adenine residue from the rRNA in the 60S subunit, and when it does this, it halts protein synthesis. Now, one special note, non-toxigenic strains of Shigella have been identified, so these are strains that do not make the toxin, and they can still invade GI mucosal cells and cause much disease. So it's believed that invasion of the mucosal cells is the main mechanism by which Shigella makes you sick. Even though it can make you sick with its toxin, that appears to be less important by virtue of the fact that non-toxic secreting cells can still cause significant disease. Enterohemorrhagic E. coli, or EHEC, also produces a toxin very similar to shigatoxin. It's called a Shiga-like toxin, and it's in the same family. It has the same mechanism of shigatoxin, and EHEC, recall, causes a bloody diarrhea just like Shigella. The classic serotype of E. coli that causes enterohemorrhagic disease is the O157H7 serotype. And one special note about this version of E. coli does not invade host cells. It attaches to intestinal epithelial cells, and people get sick from secretion of the protein into the host cells and the toxin effects. So this is different from Shigella. Shigella makes a toxin, but its main mechanism of causing disease is invasion of cells. EHEC makes a toxin, and its main mechanism of disease is the toxin because it is not invasive. Shiga toxins and Shiga-like toxin from enterohemorrhagic E. coli can stimulate cytokine release. So when they reach the systemic circulation, this is one of the things that can lead to the hemolytic uremic syndrome. And recall that enterohemorrhagic E. coli is associated with the hemolytic uremic syndrome. It's one of the worst complications of that infection that you can get. It involves acute renal failure and hemolytic anemia. And it's related to the Shiga-like toxin entering the systemic circulation and triggering an immune response. So before I go on, let me just summarize a couple key points about Shigella and EHEC. So they both produce similar toxins in the Shiga toxin family. The EHEC one is generally called a Shiga-like toxin, and they both cause bloody diarrhea. But their mechanisms are a little bit different. Shigella causes disease primarily from bacterial invasion of the mucosa. The toxin appears to be less important because strains that don't produce the toxin still make people very sick. EHEC, on the other hand, does not invade cells, so the disease is from the toxin alone and the inflammation it triggers. And then in EHEC, the toxin sometimes enters the systemic circulation and leads to the hemolytic uremic syndrome. So the next mechanism by which exotoxins work that we're going to discuss is increases in fluid secretion. And bacteria that secrete exotoxins that increase fluid secretion include enterotoxigenic or ETEC E. coli, anthrax, and Vibrio cholera. Before we talk about specific exotoxins that increase fluid secretion, let's talk in general about how fluid secretion in the GI tract is regulated. The first thing you should know is that cells in the GI tract secrete chloride, and the more chloride they secrete, the more water that is drawn into the GI tract. The next thing you should know is that secretion of chloride is controlled by the CFTR. This is the cystic fibrosis regulator protein. It's the same protein that is defective in children who have cystic fibrosis lung disease. And the way the CFTR is regulated is through a G protein. So there's a G stimulatory protein found in cells in the GI tract, and when activated it stimulates adenylate cyclase, and then adenylate cyclase increases the amount of cyclic AMP inside the cell, and that in turn activates the CFTR to secrete more chloride. So key points about this process I've summarized down here at the bottom. The amount of chloride secreted is proportional to the amount of water in the GI tract. And then if you want to increase chloride secretion, you need to either activate the G stimulatory protein or activate adenylate cyclase. And the exotoxins are going to do this to increase the chloride secretion and therefore the amount of water in the GI tract, and that's the mechanism by which they cause diarrhea. So enterotoxigenic E. coli, or ETEC, causes watery diarrhea, and it has two different toxins, and they're differentiated by their heat stability. So one is called heat labile toxin, which is not stable when it's heated up. The other is called heat stable toxin, which is stable when it's warmed to a higher temperature. So heat labile toxin activates adenylate cyclase and increases the cyclic AMP, and through that mechanism I just described 2 slides ago, it increases the water in the gut, and this is how it causes diarrhea. Heat stable toxin has a mechanism that is not as well worked out as for heat labile toxin. What we do know about it is that it activates guanylate cyclase and increases cyclic GMP. So this is different from the labile toxin, which works on adenylate cyclase. It's been shown to stimulate chloride secretion and to inhibit chloride reabsorption. And the bottom line is you end up with more electrolytes and therefore more water in the gut, and you end up with watery diarrhea. Next bacteria that makes an exotoxin that stimulates fluid secretion is Bacillus anthracus, which causes anthrax. So anthrax makes 3 proteins. The first one is called protective antigen, the second one is lethal factor, and the third one is edema factor. And interestingly, when these things are used alone, they cause no known physiologic effects, but when they're put into pairs, they produce toxicity. So one of the exotoxins that this bacteria makes is called edema toxin, and it is the protective antigen and the edema factor linked together. And it mimics adenylate cyclase, so it's going to stimulate secretion of fluid from cells. Now, anthrax causes disease in lots of different sites. The skin is most common, but also it can cause a necrotizing pneumonia in the lungs, lungs, and it can cause ulcers in the GI tract. But one thing you need to know, especially for step 1 about the skin and GI lesions, is that they often have an edematous border that may be caused by edema factor. So these anthrax lesions will often look like a black circle of necrotic skin tissue, for example, but surrounding it will be a little area of swollen skin tissue. And they may ask you this, but that swollen area is caused by edema factor and increased fluid secretion related to the exotoxin of the bug. And the last bacteria that we'll discuss which induces fluid secretion is Vibrio cholera. So cholera secretes cholera toxin, and this permanently activates that G stimulatory protein in GI cells to increase cyclic AMP, and it causes voluminous rice water diarrhea. That's the classic description of cholera diarrhea. This is common in areas with a lack of clean water, often underdeveloped countries, so people ingest contaminated water, and that's how the bacteria gets into the GI tract. And this can be a very severe illness. People can die from profound dehydration and electrolyte losses and shock if it's not treated appropriately. And actually, the mainstay of treatment is actually aggressive volume repletion. The body will clear the infection in time, but you have to give the patient enough fluid to keep up with the heavy losses caused by the cholera toxin. Now let's talk about toxins that inhibit phagocytosis, and there's really only one toxin that does this. It's pertussis toxin. So pertussis toxin is secreted by Bordetella pertussis, which is the cause of whooping cough. And pertussis toxin inhibits GI proteins, and recall that GI proteins are inhibitory. So what you're doing is inhibiting the inhibitors, and the result of that is overactivation of adenylate cyclase, and this increases the cyclic AMP levels inside neutrophils, and the result is that there is impaired recruitment of neutrophils to control the infection, and this is a way by which Bordetella pertussis persists and causes symptoms. There are two bacteria which make neurotoxins, and these are toxins that interfere with neurological function. They're both Clostridia bacteria. Clostridium tetani makes tetanus toxin, and Clostridium botulinum makes botulinum toxin. And they both work by disruption of snare proteins. So what are snare proteins? Well, recall that neurons use neurotransmitters to send signals across synapses to transmit to other cells and to muscles. And the way they use those neurotransmitters is they package them inside little things called vesicles inside the neuron. And then the vesicle docks with the end of the neuron and releases its neurotransmitter into the junction. And then it crosses the synapse and stimulates either a muscle or nerve cell. So snare proteins are present on these vesicles inside the neurons, and they allow the vesicles to dock and unload their neurotransmitter. So if a neurotoxin inhibits snare proteins, the neuron won't be able to dock the vesicles and release their neurotransmitter. So Clostridium tetani makes a exotoxin called tetanospasm, and this works on neurons in the spinal cord called Renshaw cells, and these are inhibitory interneurons. So when you inhibit the inhibitory cells, the results are that you get overactive neurological activity. This means that the muscles will always be on because you've removed inhibitory signals, and therefore you get rigid paralysis. And it's very important to remember that tetanus causes rigid paralysis. Clostridium botulinum, on the other hand, makes botulinum toxin, which works at neuromuscular junctions, and it prevents acetylcholine release. So those vesicles can't dock and release acetylcholine, and therefore you get no muscle contraction. So in contrast to tetanus, botulism causes floppy muscles and a flaccid paralysis. And I talk in detail about both of these bacteria in the module on gram-positive bugs. There are two bacteria that make toxins that lyse cell membranes. These are Clostridium perfringens and Strep pyogenes. Clostridium perfringens can cause gas gangrene when it gets into wounds, and it makes a toxin called alpha toxin, which is a phospholipase C enzyme, and it degrades substances like phosphatidylcholine and sphingomyelin. And the result of degrading these substances is that you get breakdown of the muscles, which is called myonecrosis. This toxin also causes a decline in muscle blood flow, and therefore you get occlusive plugs of platelets and white blood cells and fibrin. So the result is that you get a very low oxygen environment, which is favorable to Clostridium perfringens because it is an anaerobe. So alpha toxin helps with all of these things by lysing cell membranes and allowing the degradation of muscle cells to occur. Strep pyogenes is the causative agent of strep throat, and as you know, strep throat can sometimes lead to sequelae such as rheumatic fever and glomerulonephritis. And strep pyogenes makes an exotoxin called streptolysin O. This is also called cytolysin because it can lyse cells. And this is one of the reasons that strep pyogenes is beta hemolytic. So we talk in some of the other modules about how when you grow strep pyogenes on blood agar, it exhibits beta hemolysis. And one of the reasons for this is the presence of this enzyme streptolysin O, which can lyse cells. There's also another streptolysin called streptolysin S, and the presence of both S and O are what are responsible for the beta hemolysis. Another thing to know about streptolysin O is that there are antibodies to streptolysin O called ASO antibodies, and these are elevated following a strep infection. These can sometimes be useful if you suspect that someone has rheumatic heart disease or post-strep glomerulonephritis. The presence of these antibodies suggests they have been exposed to strep pyogenes. You should know that toxins are the basis of many vaccines, and the way we use a toxin as a vaccine is to turn it into a toxoid. So a toxoid is an inactivated bacterial toxin that won't cause patients to become as sick as the toxin does, and you can inject that and create an immune response, and this is a way of vaccinating people against infection. And two of the main bacteria for which this is used is to prevent diphtheria and tetanus. So there's a vaccine called the DTaP, and this stands for diphtheria, tetanus, and acellular pertussis. So it's a toxoid to diphtheria, it's a toxoid to tetanus, and it's an acellular pertussis. So it's an inactivated toxin plus some bacterial elements all mixed together to immunize people against pertussis, which, recall, causes whooping cough. Let me make a comment on the genes that bacteria use in order to create these exotoxin proteins. You should know that many exotoxin genes are not part of the bacterial chromosome. Some of them are encoded on a plasmid, which is a small section of DNA that's separate from the chromosome. For example, E. coli heat labile toxin is plasma encoded. And then many are encoded by a bacteriophage. So a bacteriophage is somewhat like a virus, and it delivers a little bit of DNA to a bacteria and then gives it a new ability. And one of the classic bacteria that uses a bacteriophage to acquire its ability to make exotoxins is Corynebacterium diphtheriae. Bacteriophage encoded toxins also include strep pyogenes, erythrogenic toxin, E. coli Shiga-like toxin, botulinum toxin, and cholera toxin. Probably the most important one to remember is this one right here. Diphtheria uses a bacteriophage to acquire the ability to synthesize its exotoxin. The last topic I'll cover in this module is that of superantigens. So superantigens are exotoxins that cause a massive number of T cells to be activated. So let's review how a normal antigen is processed and presented to the immune system. So normally an antigen presenting cell takes a bit of an antigen and sticks it on top of an MHC complex, and then that triggers a T cell receptor and activation of a single T cell. What superantigens do is they do not get processed. They directly connect the MHC complex to the T cell receptor on a T cell, and they do this indiscriminately, and as a result they activate many more T cells than would normally be activated, and they trigger a massive immune system response. So to put this into perspective, a typical antigen activates less than 1% of T cells. A superantigen can activate 2 to 20% of T cells. This results in a huge release of cytokines like interferon gamma and IL-2 and massive vasodilation and shock. So bacteria that possess superantigens can make patients very sick. There are two bacteria that synthesize toxins that can act as superantigens. The first one is Staph aureus. Staph aureus makes toxic shock syndrome toxin, or TSST1, which is a superantigen. Strep pyogenes, which is also called group A strep, also synthesizes a superantigen. It's called pyrogenic exotoxin, either A or C. Both of these bacteria can cause the toxic shock syndrome, which is the body's reaction to a superantigen. It's much more common with staph. Classically, after a woman has had a tampon in her vagina, there's staph present in the vagina normally, but for reasons we don't understand, sometimes it develops the ability to synthesize these superantigens, and it can cause the toxic shock syndrome. There is something called the strep toxic shock syndrome, but this is rare, and when patients have it, it's usually associated with necrotizing fasciitis, which is a very serious skin infection. On board questions, if they describe the toxic shock syndrome, they are most likely talking about staph. So let's briefly review what the toxic shock syndrome is. It's fever and shock, which is hypotension, and a red rash diffusely over the body. Patients have this diffuse red erythroderma. It looks like a sunburn, and after a few weeks, eventually the skin will start to desquamate and fall off on the palms and soles. One thing that's very unique is that diarrhea is common to the toxic shock syndrome. So when you see this in a question, it's often going to look like someone who has sepsis. They're going to have fever and shock. But the two things that will tip you off will be the diffuse sunburn type rash all over the body and the presence of diarrhea. When you see those two things, they're probably describing toxic shock syndrome and not not sepsis. Multi-organ system failure often results, and this is a life-threatening infection. And classic scenarios are staphylococcus plus some type of packing material where it can grow. So women who've had a tampon inserted or surgical wounds that are packed, both of these things can trigger someone to develop the toxic shock syndrome. And that concludes our module on virulence.